Alright guys, so in this video I'm going to provide a very useful overview of the steps involved in RNA-seq and ChIP-seq analysis. You can also apply the same steps to a number of other bioinformatics analysis like DNase I-seq or ATAC-seq, etc. I'm also going to tell you about the tools or softwares that are commonly used in the bioinformatics analysis. So let's get started. What you get out of the sequencer, for example, say you're using Illumina, is a fastq.gz file. And this is just a zipped fastq file. The first step is to unzip it and convert it into a fastq file. And I already have a video on how to do that. It's a very simple step. You can easily do it. Also, while we are on the subject of fastq.gz files, depending on how many reads you have in your sample, the sequencer might spit out a number of fastq.gz files for one sample. Say if a lot of signs, 4 files per sample, and you can just concatenate all of those into one and then unzip and convert it into fastq files. Okay, so now you have your fastq files. What do you do next? Next is the quality control, and this quality control I most often use fastqc. That's the name of the program, and there are a number of other programs available as well. So why do you have to do quality control, and what the output of fastqc looks like? So here is one of the output of fastqc. There are a number of them. I have made another video on it, and be sure to check it out on my channel. So the quality score of all the bases is represented in it like this, at least in the fastqc result. So on the x-axis here, you can see it's the position in read in terms of base pairs. So say you got your sequence and you asked the sequencer to read 100 base pairs of your reads. So this each segment here represent one base pair, and all the way up to 99. And the y-axis is the Phred score. It's just the probability of the error. So higher the Phred score, lower the probability of error, and it's usually represented in a box plot format. So the general rule of thumb is that anything above 30 is really good. And you can find out, if you just Google the Phred score, you can find a lot more detail about what these number actually means. And so this, you can see that this graph is divided into 3 colors: green, yellow, and pink or red here. And you're usually okay if you are anywhere above, say, 28 or 30. That's a good quality score. Anything in the yellow zone, you're now seeing lower quality scores, and red zone, you really want to get rid of it. So this is a normal trend in the Illumina sequencer. And also here, the blue line is actually the average score that you're seeing. So this is a very common trend of Illumina sequencers. It could be HiSeq or MiSeq or NovaSeq. As you go towards the end, say you lose the quality of your reads or bases. So a lot of times you need to trim off. So for example, in this case, to be safe, I would trim last 40 base pairs and only keep up to 60, and then use those 60 base pairs in the reads for the further analysis, like alignment. Okay, so going back to our chart here, so you are doing quality control using fastqc trimming if necessary if your reads are the quality is not good towards the end, or a lot of times your reads have adapter contamination. If your primers are reading too far, then you can also trim the adapters off. And there's another program or software available for trimming. It's called FastX Trimmer that I use a lot, and it's very easy. Once you are done with the quality control, you can also count the number of reads. At this stage, using FASTQ files, and also split the FASTQ file if necessary. If you have too many reads in your file, that will take a lot of time for mapping. Say, if you have more than 50 million or 100 million, I would usually split the FASTQ file into two and then try to map individually, and then later combine the mapped files. Okay, so once you have done the FastQC and trimmed if necessary, the next step is to map the reads on the genome. Now, there are two most common programs used for mapping the reads on the reference genome. One is Bowtie2 that I would recommend that using for the ChIP-seq analysis, and HiSat2, which is really fast in my experience, at least 10 times faster than Bowtie2, preferred for RNA-seq analysis. And the reason is that HiSat2 is a splice-aware program. And if you have splice variants, and if you're in general doing RNA-seq analysis, just do the mapping using HiSat2. So just to give you a brief overview of what is mapping or alignment, say you are doing RNA-seq analysis, you have a bunch of reads produced by your sequencer, and now you used, say, for RNA-seq HiSat2 to align it on the genome. So you have a reference genome, and you are just physically allocating using the base complementarity your reads on the reference genome. And as I was saying, that HiSat2 is splice-aware program. It would take care of any introns that are actually, if your sequence is, it involves two different exons with the exon, it will actually take care of that. So simply stating, the mapping or alignment is just putting your raw reads onto the reference genome. All right, so once you are done with your alignment, this both HiSat2 and Bowtie will generate SAM files. Okay, and next step is to convert this SAM file into the BAM file. Don't worry about the names. Just remember for now that the mapped file is SAM file, and it's usually converted into BAM file using SAM tools. Now, why do you need to convert the SAM file into BAM file? Simple. SAM files are very big in terms of size, and we can read them. You can open it and read what it looks like, what's in there. But since they are really big, to save some space, you convert them into BAM file, and BAM files are much smaller because they are binary file. Even if you open them, you cannot read. You just would see a bunch of random alphabets and numbers in there. The reason is just it's a binary file, and it's very smaller in size. And since space is always a problem, you always convert the SAM file into BAM file. And the program that's used for this purpose is SAM tools. Once you have converted into BAM file, which is much smaller, you can index it also using SAM tools, and then convert. What I usually do is always convert it into what's called BigWig files. So don't worry about all these file names. They have nothing to do with your actual this is just computational complexity. You don't have to worry about why they are named as SAM or BAM or Bigwig. They're just people who developed these files, just gave them specific names based on some of the characteristic features of those files. But as far as biology is concerned, it means nothing. Now, once you convert your BAM file into Bigwig file, you don't necessarily have to, but if you convert your BAM file into Bigwig file, you can visualize it in IGV, that is Integrated Genome Viewer. And this can be done using deep tools. And I really like to do it for this reason. So say here I'm just showing you an example. You have a number of samples, and this is a snapshot of IGV, and you can literally see after you normalize it for the count per million. So for example, in case of this gene here, I have some ChIP-seq result. In these two cases, the particular protein binds, but in these two mutants, it doesn't bind. Or in case of, if you have I have these 4 panels of RNA-seq analysis, and you can see in this particular mutant, the expression of this particular gene is low. So it's a very good visualization technique. So I personally, for my all of my analysis, I convert BAM file into Bigwig file and try to look at in IGV and see what's the pattern. So once you have done there, then you can do the gene counting using BET tools. And gene counting is just now counting how many reads are actually coming from each gene, and that will give you the gene expression. And then after that, you just put those gene counts into EDGAR or DNase programs, which are R-based. So all of this analysis is going to be in your, say, your own computer in the terminal or supercomputer or server. And once you have the gene counts, you can actually just do the further analysis, differentially expressed gene or DEG analysis in R. Okay, so that's the overview of the RNA-seq and ChIP-seq analysis. A lot of things, for example, how to convert fastq.gz file into fastq file, or do the quality control. I have made separate videos on my channel. If you will like, make sure you check those out and let me know if you would like me to make more detailed video on any of these other steps. Thank you. Hope this was informative.